Hello and welcome to the series finale for Pesticide History. In this episode, we'll cover biopesticides, their mechanisms and history. Let's begin. The first one we'll cover is Bt. After World War II, a bacteria called Bacillus thuringiensis, or Bt, was discovered. It was found in a diseased larvae in the Mediterranean flower moth. It had limited viability, however, as effective pesticides simply because the synthetic pesticides worked a lot better, although it found use in genetic modification. Narcological control was the fungus called Horazium. The species M. arachium was found to be effective in controlling locusts in Africa, and could be used in a low-tech spray equipment method by mixing it with vegetable oil and using an ULV spray technology. This species of fungus was also used to deal with grasshoppers in Australia, and another species discovered was used to treat vine weevils on strawberries and other soft fruit crops. Bt likewise was developed further, with several subspecies being found that could be used to control different types of caterpillar pests. In later days, the genes then code for the proteins that are used by the fungus to kill different types of caterpillars were incorporated into plants via genetic engineering, allowing the plant to directly kill their pests. Another biocontrol is Bavaria bassiana. The biopesticide is used for greenhouses to control whitefly. It's once again a fungus, and its fungal species feeds on most insects. There have also been attempts to culture viruses to deal with insect pests, such as bacilloviruses. These biopesticides, however, are very expensive due to the need of living insects to culture the virus in, although a few species of this virus are available for use to deal with codling moth, gypsy moth, leafworm, and cassava hornworm. Now onto biofungicides. There are a litany of microbe species being tested out to deal with various species of fungus that are soil-borne, some of which have been released to public use. The first one we'll cover is Conanrifrum minetans. This is the only biocontrol agent registered in Germany as a biopesticide. This is a mycoparasite that consumes white mold. It was initially tested in England and was found to consume the fungus on a number of different substrates such as barley and wheat. And this mycoparasite infects 80% of the white mold it comes in contact with. The next one on our list would be Glaucladam Virons. This species was found to be able to control Pythium and Rhizoctonia pathogens in soilless mixes within greenhouses. It was cultured by the USDA in the late 1980s. It was first discovered in a soil sample in Maryland. This fungus produces as a waste product the toxic compounds glyovirin and glyotoxin, both of which are very toxic to Pythium and Rhizoctonia. In essence, the fungus's poop is poisonous to other fungi. The next species on our hit list is Tritoderma Horazium, strain T22. The base species parasitizes different fungal species, although those that dwell near roots do not consume as many fungi as the ones found in wild soil. Strain T22 was made by fusing the cells of T95, a strain of the species that's found in the root system of different plants, and the strain called T12, found in just regular soil. The result of this fusion allowed for a strain of this parasite that can survive and thrive and mass produce in the root system of plants while rapidly consuming any other fungal species around. It's commonly used as the seed treatment known as root shield, which provides control equal to fungicides when applied as a seed treatment. As a preventative, it is commonly used in various ornamentals like poinsettia, geranium, and impatiens. It also competes with other colonizing fungi and bacteria, and also causes an induced resistance to different fungi within its host plant. Now on to Streptomyces geoverdis, known by the trade name Mysostop. This is a parasite that was found in sphagnum peat moss, and became researched and eventually found to be useful for controlling Fusarium wilt in basil and carnations and Douglas fir. Next on our hit list will be Glaucladam Virons. This species was found in Finnish field soil and was used as the active ingredient in Primostop. It seems to be effective against different types of root pathogens such as damping off, seed rot, and root rot. As of right now, it's used mostly in ornamental bedding plants, although effectiveness varies. The last one we will cover is non-pathogenic Fusarium Oxysporum, strain FO47. Initially found in a soil that had no Fusarium infections in France, it was discovered that that landmass had a strain of Fusarium that rapidly inoculated the plant but did not cause any damage. Basically, it's a fast-growing, asymptomatic variant of Fusarium. This was then cultured out and used to treat different types of diseases as a preventative within greenhouses, as well as outdoor tomato and ornamental growth. The main mechanisms of action include the induction of host defenses and direct competition of other strains of Fusarium. This strain was found in that soil, as I mentioned before. This landmass was studied for over 25 years by the organization INRA, and this Fusarium species is a culmination of that 25 years of work. Well, that about covers everything. Thank you for watching. Thank you for watching this video. If you're watching on YouTube, please subscribe to me on BitChute for a greater variety of content, 4 videos a week. And thank you to all my subscribers on both platforms. I appreciate it.